Department of Genetics here is well over 100 years old. Its first professor of the Arthur Balfour Professor of Genetics was Reginald Punnett of Punnett Square fame. At the end of the 50s, the university was looking for a new chair of the genetics department, and a man named John Thody was eventually appointed by the Vice-Chancellor as the Arthur Balfour Professor of Genetics. John had come from Birmingham, where he began to use fruit flies to look at natural variation, in terms of the number of bristles on the fly's back and how that varies in natural populations. And that's an area of genetics, sort of quantitative genetics, trying to understand how contributions from many different genes affect these quantitative traits. And it's very useful for things like plant breeding. Anyway, John became head of department then until the mid-80s, I think John retired. But he not only expanded the Department of Genetics, which in Cambridge was viewed as a bit of an upstart science, expanded the teaching of genetics in the undergraduate curriculum, but more importantly established Drosophila as a research organism in the department. And then I think the next most powerful part was Michael Ashburner, who I actually came here to postdoc with in 1990. Michael was an undergraduate here in the early 60s, and in 1964 started a PhD in the department under the guidance of Ian Henn, looking at the chromosomes in the salivary glands of maggots. It's not quite important why he was doing that, but it gave him very powerful insights prior to the whole advent of molecular biology as to how gene control circuits worked. And Michael was very influential and quickly became super prominent in the world of Drosophila genetics. He was considered one of the father figures. I'm surrounded by books that Michael Ashburner wrote on Drosophila. He was one of the world's leading experts, and he recruited a lot of people to Cambridge in general. People were attracted here because of Michael's influence. And furthermore, Michael was one of the very early adopters of molecular cloning in the 70s, where one took bits of DNA from somewhere else and grew them up in bacteria under very strict conditions at the time. But Michael's influence cannot be underestimated in terms of the support of Drosophila as a research organism and collecting huge amounts of information on its natural history. But I think equally as important, he was an early champion of bioinformatics and genome biology. He was involved in the efforts to sequence the Drosophila genome, which was done in 2000, the second animal genome to be sequenced, but more importantly involved in building up computer databases, so transferring this big book into the sort of website interface Flybase that we have now for interrogating Drosophila. So since, although John Thody started it, it was Michael that was really dominant in developing Drosophila research here, and that's why I came to the department.